My name is ANISSA ABI-DARGHAM, and I'm a professor of psychiatry and radiology at Columbia University and New York State Psychiatric Institute. So I use brain imaging tools to examine the neurochemistry and the function in the brains of patients with schizophrenia and other disorders, and sometimes comorbidity. Dopamine is very involved in schizophrenia and multiple aspects of the disorder and multiple symptom domains. So patients with schizophrenia have psychosis, that is the hallucinations, delusions, and thought disorder. They also have other symptoms, the cognitive deficits and negative symptoms, which are more the withdrawal and the flatness of affect, poverty of speech, poverty of thoughts. And what we've been able to characterize so far very well is the fact that excess dopamine and excess stimulation of one of the dopaminergic receptors, the D2 receptor, in one part of the brain and the striatum, that those are associated with the psychosis elements of the disease. What's been very difficult to do is to examine other parts of the brain, which are very important. For example, the frontal cortex is involved in cognition and our ability to think, memorize, strategize, take actions, change our behavior, adapt to the environment. And dopamine in the frontal cortex is essential for these behaviors. It's been difficult to get at examining dopamine in the frontal cortex because of the technicalities, because the way the tools we use for imaging necessitate being able to visualize the D2 receptor and then provoking kind of a change in dopamine to induce a competition for binding of a PET radiotracer to that D2 receptor. And because D2 receptors are in small amounts in the frontal cortex, it was difficult to pick up that signal. Until very recently, a new method was developed, and we adapted this method. We used a new PET radiotracer with a psychostimulant challenge, and we were able to address the issue of what about dopamine outside of the striatum. And so this is the study that we're reporting on now. And what we found is that there are profound deficits in the capacity for dopamine release, so the availability of dopamine. The availability of the dopamine neurochemical in the cortex in patients with schizophrenia. But it wasn't just restricted to the cortex. It seemed to affect almost every other brain region outside of the striatum, including the brain region where the dopamine cell bodies are. So this is where dopamine cell bodies are, and they project from there throughout the brain. So in that region where is housing the dopamine cell bodies, there's also a deficit in dopamine release capacity, which means there's lack of availability of dopamine pretty much throughout the brain in schizophrenia, with this exception of the striatum, where there is a little too much. So the new finding is that there is a dopamine deficit in most of the brain outside of the striatum, including the frontal cortex and the midbrain, where dopamine cells originate. So there are multiple levels of significance. The first one is just the basic essentials of understanding the neurochemistry, the alterations, mapping the abnormalities so that we can understand the disease, and that helps us to design studies to first address the alteration, treatment studies, or to understand mechanistically how does an alteration as such emerges in the brain. So how is it that we can have excess in one region and a deficit in another region? This is a paradox that we have to address that wasn't documented before, wasn't known before, was suspected, but this is the first documentation of it. So how is it mechanistically is that one dopaminergic projection is overactive, such as in the striatum, and all the others are underactive? Or is it that it's overactive in the striatum because of the local regulation within the striatum? So it's a locally driven excess and not something that is coming from the midbrain dopamine cells themselves. That's the first significance, is to just helps us to think about the disease mechanistically and to design studies to address the mechanisms. The second level of significance is treatment, of course. First, it helps us to understand why our antipsychotics, which essentially all are D2 antagonists, and that's how they work, why these are so little effective at treating the non-psychotic symptoms of the disorder. And that is because these symptoms, which originate in the other parts of the brain, are not really related to excess dopamine or excess D2 stimulation. It's the opposite. They're due possibly to a deficit in dopamine. I think that in the immediacy of it, there isn't really a direct meaning to the patient. It's more of a long-term meaning, which is a better understanding of how some of the symptoms they're suffering from are emerging, what they're related to, and how to better treat them. So the next step is to try to address this alteration that we have now documented, improve dopamine transmission in those brain regions, design studies that can address at the same time the deficit in the cortex while treating the origin of the psychotic symptoms in the other parts of the brain. What we're looking at in this figure is a plot of the PET data on the left side of the screen, the PET and the fMRI data combined on the right side of the screen. In blue, the blue dots represent the healthy volunteers that participated in the research, and the triangles, orange and blue, are the patients with schizophrenia. So on the left side of the screen, we're looking at how much dopamine is released, our estimate of how much dopamine is released via our PET paradigm. So the bigger the number from 0 up, the more dopamine is released. And we can see that the controls release on average close to 10%, while patients with schizophrenia do not release at all. They're actually below 0. So this is the evidence showing that patients with schizophrenia do not release dopamine in response to this paradigm the way healthy volunteers do. Now we try to understand how this inability to release dopamine, or the capacity of subjects to release dopamine, relates to their capacity to activate their frontal cortex in response to a working memory task, the demands of a working memory task. And what we saw is that in both groups combined, the higher the dopamine release, as seen on these lines going up, the better they were able to activate their prefrontal cortex in response to the working memory challenge. So more dopamine release capacity, better activation, better frontal cortical function. I want to thank the patients and the families that have participated in this research, because without their participation, we cannot advance our understanding of the disease.